Hello and welcome all for this continued lecture series of enzymology. So today we will discuss, today is our session 38 and the topic is enzyme inhibition. Yeah guys, we have been discussing about what are enzymes, the classification of enzymes, and we have also discussed the properties of enzymes, and we have also discussed the kinetics of enzymes, and we have also discussed the mechanism of enzyme action. Now in today's lecture we'll focus on what is enzyme inhibition, and before going to what is enzyme inhibition, we will try to define what are inhibitors. Inhibitors are compounds uh, that decrease an enzyme's activity. So inhibitors are compounds, they may bind somewhere to the enzyme. Sometimes they bind to the active site, and sometimes they bind other sites, and somehow they are going to inhibit the activity of the enzyme. So the enzyme cannot further convert the substrate into the product. So such compounds are called as inhibitors. They are going to decrease the activity of an enzymes. So the inhibitors can be irreversible inhibitors, reversible inhibitors. So reversible inhibitors are those they bind to the enzyme and they bring down the activity, and they can also dissociate from the enzyme after some time. They are called as reversible. The process is reversible. And irreversible inhibitors are some compounds. Once they bind to the enzyme, they completely shut the activity of the enzyme molecule. They are usually powerful toxins. Sometimes they are being used as drugs. Yeah. Now let us look into the type of the uh, enzyme inhibition. So enzyme inhibition can be of three types. One is competitive inhibition, and then we have uncompetitive inhibition, and then we have non-competitive inhibition. Uh, so in competitive inhibition, so basically we have irreversible inhibitors and reversible inhibitors. So in reversible inhibitors, we have two types. Here there are two options, two possibilities. The free enzyme, the reversible inhibitor can bind to the free enzyme and prevent the binding of the substrate. The enzyme substrate complex, the reversible inhibitor can bind to the ES complex and prevent the reaction for the conversion of the product. Now let us see that in detail. So in competitive inhibition, there is a competition with substrate. Here the substrate competes for binding with the active site and does not affect the catalysis. So this is a typical uh, this is a typical this is a typical enzyme substrate catalyzed reaction. Yeah, let me take this. This is a typical enzyme substrate catalyzed reaction. So where we represent this as enzyme plus substrate gives rise to enzyme substrate complex, and which in turn gives into enzyme plus product. Then when we have enzyme plus inhibitor, so it forms an here there is a competition for the active site of the enzyme between the substrate and the inhibitor. So sometimes it may form EI complex, sometimes it may form EA complex. So if EA complex is formed, we can't expect the product to form. We can't expect the product to form here. So here the kinetics are going to change. So this is the schemate. So this is an enzyme and this is the active site of the enzyme, and so this is reversible. The substrate can bind and you get an enzyme substrate complex, or in presence of inhibitor, the inhibitor may go and bind to the enzyme and it may form EI complex. So it may form EI complex, and both the reactions are reversible. So the kinetic point of view, what are the kinetic parameters that are going to change? So there is no change in the Vmax, and there is apparent increase in the Km. So Km is going to increase, and Vmax, and there is no change in the Vmax, and the LB plot lines intersect at the y-axis. Now let us look into this. Yeah, so this is the kinetic LB plot of competitive inhibition. So there is no change in the Vmax, but the Km is going to increase. See, here we can see the Km is from here 1, 2, 3, we can see different. The Km is going to increase, and the there is no change in the Vmax. And uncompetitive inhibition, so in uncompetitive inhibition, only binds to the enzyme substrate complex. The inhibitor here, it binds to the enzyme substrate complex. So in the previous one we saw there was a competition between the substrate and the inhibitor to bind to the active site. So it does not affect the substrate binding, inhibits catalytic function. So now let us see this. Let me annotate this. Let me take red. Okay, so this is a typical enzyme substrate catalyzed reaction where an enzyme, in absence of an inhibitor, a substrate molecule binds with the enzyme, forms an ES complex, and it converts it into E plus P. So in presence of inhibitor, now you look at where the enzyme is going to bind. Where the inhibitor is going to bind. The inhibitor is not binding to the enzyme. It is not competing with the enzyme. Rather, the inhibitor is going to bind to the ES complex. So this is very, very important. So hence it forms ESI complex. It forms ESI complex. So this is a typical enzyme, and you have substrate. The substrate is binding in the particular pocket, active site, and you have another pocket where the inhibitor is binding. It forms an ESI complex. It forms an ESI complex. So what are the kinetic changes that we are going to observe from the normal Michael-Mentenes kinetics? So here, in presence of an uncompetitive inhibitor, there is decrease in Vmax. So there is decrease in Vmax. So in case of competitive inhibition, there was no change in Vmax. There was no change in Vmax, and there is apparent decrease in Km. So there is decrease in Km also, but their Km was increasing. So here their Vmax was constant and Km was increasing, and here Vmax is also decreasing and Km is also decreasing. So no change in Km bar Vmax. And Lineweaver lines are parallel. So there we saw the intersection on the y-axis in case of competitive inhibition, and here here you can see the parallel lines are parallel here in case of uncompetitive inhibition. So a small application of enzyme inhibitors. Many enzyme inhibitors can serve a variety of purposes, including medicine to treat diseases, agriculture as pesticides. For example, the competitive inhibitors in the treatment of influenza via the neuraminidase inhibitor Lenenza, which is marketed. An example of a use of non-competitive inhibitors is the use of cyanide as a poison, prevents aerobic respiration. So many drugs are being used as competitive, and many enzyme inhibitors are being used as drug, and the research is going on even till today. So this is the overall summary or the sum up of today's presentation: enzyme inhibition, different modes of inhibition. These are called as double reciprocal plots, or sometimes we call it as an LB plots. A LB plot is nothing but when we take the reciprocal of the Michael-Mentenes equation. So what Michael-Mentenes equation we have derived, when we take 1 upon the MM equation, so we get the LB plot, so that graphically we can represent it. Hence we have to transform the MM equation into the LB plot. So this is in case of a competitive inhibitor. So in competitive inhibition we can see the intersection here. So this is when we take so in MM equation we have taken S versus V versus S. So here it becomes 1 by V versus 1 by S, because we are taking the reciprocal. So in case of competitive inhibition, you can see the intersection here, and in case of uncompetitive inhibition we can expect parallel lines. So here there is a comparison. So in absence of inhibitor, no inhibitor present, the Vmax is increasing. In presence of inhibitor, we can see here there is a there is no change in Vmax, but there is change in the increase in the Km. There is increase in the Km, substrate concentration, right? In case of uncompetitive inhibition, we can see the comparison. In absence of inhibitor and in presence of inhibitor, we can see the lines are parallel, they are not meeting. And in a non-competitive inhibition. So when there is no inhibitor, we can see there is no change in the Vmax. But when we come to the non-competitive inhibitor, here we can see the junction meeting of this both of these lines on the x-axis. On the x-axis we can see it meeting the point on the x-axis, where x is equal to -1 by km. So here it's a overall summary: type of inhibition with respect to Vmax with respect to inhibitor, Km with respect to inhibitor. In competitive inhibition, Vmax stays the same, Km is increased. Non-competitive inhibition, Vmax is decreased, Km stays the same, it remains same. In uncompetitive inhibition, Vmax is decreased and Km is also decreased. So this is all about the inhibitors and the role of inhibitors in enzyme kinetics, and lot of research is going on in discovering newer and newer enzyme inhibitors which can bind reversibly and permanently to the inhibitor to the enzyme active sites of the proteins and active sites of the enzymes, which can in turn act as future medicines. So thank you one and all for the lecture. I hope you understood. If there are any queries or questions, please post it in the comment box, or you can post it in my Google Classroom. Thank you. Have a stay safe, maintain social distance. Thank you one and all.